Biotechnology. In this chapter, we would study some important terms like genomic library, polymerase chain reaction, DNA fingerprinting, gene sequence, gene therapy, tissue culture technique, and much more. Now, what is biotechnology? The molecular genetics which enables us to manipulate genetic material for the welfare of mankind. As we all know, scientists all over the world isolate genes that have some fault in them and then insert the normal genes in them. Biotechnology is solemnly done for the benefit of human race. Desired varieties are formed by gene recombination. Now, what is gene recombination? Gene recombination is the insertion of a foreign gene into the DNA. A gene with specific speciality is isolated and then inserted into the DNA, and the organism is allowed to reproduce. At the development stage, it would develop the desired varieties. The recombinant genes are made for the production of substances such as enzymes. In the case of enzymes, vitamin B3 is made when three enzymes are aligned up with a specific amino sequence. Antibiotics, which are used in the immune system, hormones, as insulin is only made by the field of biotechnology needed for the human use. Now, there are some special terms used in biotechnology. Cloning, it is the development of identical cells or organisms. In this process, nucleus from the zygotic cell is removed and another nucleus from the somatic cell is inserted in its place. After the organism is allowed to develop, the new organism is now would be identical to the first one. Recombinants. Formation of the new association of DNA or the insertion of a foreign gene into a DNA is known as recombination, and the DNA is known as recombinant DNA. Now, how this foreign gene would be inserted into the DNA? It would be done by carriers known as vectors. A vector could be plasmid, which is bacteria, or phage, which could be virus, that inserts the piece of new or foreign gene into the DNA. Gene, it is the unit of inheritance. Genome is the genetic material in an organism. Liposome, a lipoprotein sac which fuses with the host cell and insert or introduce the DNA or protein. That lipoprotein sac is known as liposome. Gene cloning, identical copies of genes are produced. PCR, polymerase chain reaction. In this reaction, number of gene copies are produced within a test tube. Reverse transcriptase, RNA directed DNA polymerase which can make DNA strands complementary to RNA. Any such DNA strands are made which are complementary to RNA.